everyone. We would be discussing a small topic from biochemistry today, that is proteins and their synthesis. Proteins are the most abundant organic molecule present in the living organism. They constitute about 50% of the dry weight of the cell. The basic structural units of proteins are mainly amino acids. So why are they called as amino acids? Because they consist of an amino group and a carboxylic group. See, this is a structure of amino acid which consists of an amino group and a carboxylic group, and hence they are called as amino acid. We have a side chain called as R. So based on the differences in the side chain, amino acids can be classified into aliphatic amino acid, aromatic amino acid, sulfur containing, basic, acidic, etc. So the side chain, the differences in the side chain is what determines the structure of the amino acid. Proteins can be broadly classified into simple, conjugated and derived based on their chemical nature. So what is meant by simple proteins? Simple proteins means they just have the amino acids arranged by peptide bonds to form a polypeptide chain. Examples of simple proteins include albumin, globulin, elastin, keratin, etc. While conjugated proteins are wherein the simple proteins are combined along with other non-protein parts such as a prosthetic group. So the examples of conjugated proteins include hemoglobin, DNA, RNA, etc. While derived proteins are mainly formed as a result of denaturation of proteins as well as degradation of proteins to form coagulated proteins, peptides, peptones. So all these constitute the derived proteins. Now, let us have a look at how the protein synthesis occurs. So the basis for protein synthesis is mainly the central dogma of molecular biology, wherein the genetic message within the DNA is first transferred onto an RNA, and based on the message in the RNA, corresponding proteins are synthesized. So DNA to RNA and then to protein is what constitutes the central dogma of molecular biology. See, protein synthesis mainly occurs in two steps, that is transcription and translation. So transcription mainly is the conversion of the message within the DNA onto an RNA is what is transcription, whereas synthesis of a protein based on the message in the RNA is called as translation. But I would say protein synthesis can be broadly divided into four steps, that is transcription followed by post-transcriptional modification and translation followed by post-translational modifications. So transcription is a process by which a base sequence of mRNA is synthesized on a template of complementary DNA, whereas in translation, the process of synthesis of proteins in the ribosome using an mRNA is called as translation. So before we move on to the steps in protein synthesis, we can have a look at the different types of RNAs. So one is mRNA or the messenger RNA, which is synthesized in the nucleus. It carries the transcripted information from the DNA. So that is the role of mRNA. While tRNA is called as transfer RNA, which mainly transports the information to the site of protein synthesis. tRNA is said to be having a clover leaf shape. rRNA is ribosomal RNA, which is synthesized in the nucleus, and they act as factories of protein synthesis. So now the first step, that is transcription, it is divided into three sub-steps, that is initiation, elongation and termination. So for initiation of transcription, the RNA polymerase II enzyme should bind on the promoter site of the DNA double helix. So examples of promoter sites include catbox, homeobox, etc. So the RNA polymerase II enzyme binds onto the promoter site on the DNA double helix, and that initiates transcription. So what occurs? What happens? Actually, the DNA unwinds, okay, and then you can see the nucleotide bases which are actually opened up. Now the mRNA synthesis starts. So that is what is the process of elongation. Usually for transcription, the 3' 5' strand of the DNA act as the template strand, and hence the mRNA that is the complementary mRNA form, would be having a 5' 3' direction. Okay, so that means the 5' end of the mRNA constitute its head, while the 3' end constitute its tail. So when the mRNA synthesis occurs, when it encounters any stop sequence there, the transcription process gets terminated and the mRNA is released. Okay, so once whatever mRNA formed just after the termination of transcription is called as pre-mRNA or the primary transcript. Now it will enter onto the post-transcriptional modifications for it to become the final mRNA. So the three main post-transcriptional modifications are splicing, a cap addition and a poly-A tail addition. So what is meant by splicing? In the pre-mRNA, you will have various areas that is which are non-coding in nature. Those are called as introns. So splicing is the removal of introns and joining of the exons together. That is called as splicing. Now onto that spliced mRNA, we should add a cap at the 5' end and a poly-A tail. That means multiple adenosine units are added onto tail end, that is the 3' end of the mRNA. So these are mainly done to make the mRNA much more stable and to prevent its dissolution by various body enzymes. So these are the post-transcriptional modifications. Once the post-transcriptional modifications are done, then that comes the final mRNA. So the transcription as well as the post-transcriptional modifications occur within the nucleus, and once the final mRNA is formed, it moves on to the cytoplasm towards the ribosome. So that is where the translation, that is protein synthesis, occurs. So the picture shows the mRNA which is attached onto the ribosome, and it has the triplet nucleotide bases which constitute the codons. Now the tRNA will be having the corresponding anticodons in it, and it will go to the amino acid pool to take up the corresponding amino acid, and it comes and momentarily binds with the codons in the mRNA. Okay, now the amino acids are joined together by means of peptide bonds, which is brought about by the rRNA, that is ribosomal RNA. Once the amino acids are joined together to form the peptide bonds and thus the polypeptide chain, this tRNA breaks the contact and moves away. Okay, that is how the polypeptide chain, that is the protein, is synthesized. So once the translation is done, the protein formed would be having the primary structure, that is, it will just have the amino acids attached by means of the peptide bonds. So that constitutes the primary structure of protein. So what is meant by the secondary structure of protein? Wherein surplus hydrogen bonds act to create an alpha helix and a beta plated sheet. So alpha helix and the beta plated sheet forms the secondary structure of protein. And then other bonds, whereas like disulfide bonds, covalent bonds, ionic bonds, etc. would act on the secondary structure to produce a tertiary structure, while in quaternary structure more than one polypeptide chain is usually involved. So post-transcriptional modification mainly concentrate on making the protein structure more complex, that is, from the primary structure. During post-translational modifications which occurs in the Golgi apparatus, the protein would conform onto the tertiary and quaternary structures. So for the protein to be having a biological activity, it should at least have a tertiary or quaternary conformation. So that is how the proteins are synthesized and modified to meet the body needs. So that completes the synthesis of proteins. Thank you.